and then usually this is all covered with septum pellucidum, and that just depends on how accurate I am with cutting a pure mid-sagittal section. Apparently at this point not so great. But the piece that you have to kind of focus on is that here's the diencephalon, and as the fornix comes curving over the top, here's the interventricular foramen. So this is how CSF gets from the lateral ventricle into the third ventricle. And remember, that's the point that I'm going to use as a landmark, because that interventricular foramen, when we cut through this, is going to mark a number of different landmarks. It's going to be at the anterior pole of the diencephalon, or the anterior pole of the thalamus. When we get into it, it's going to be at the genu of the internal capsule, where the internal capsule comes all the way to the ventricular system. It's going to be, when we look at it in a coronal section, it's going to ultimately cut through the rostral part of the temporal lobe, which will be the amygdala. So that interventricular foramen is really our landmark that gives us the best access to see all the different structures that we've talked about in class, okay? So that being said, I'm going to take a brain, and cut through it. And I'm going to try to be as close to midline as I can. And then we open it up, and now we see, here's that similar view. Here's corpus callosum. This time the fornix is complete, so you can run it around, and here's that interventricular foramen. On the other side, here's the fornix coming around. This side's torn up a little bit. If you notice, kind of there's a track like that. That's where they put, where those skulls had the little wooden plug in them, and they stick a large needle in there to infuse a lot of extra fixative in there. So that's why it's torn up. Okay, but that's where that is. But here again is the interventricular foramen. Here on this side, you can see kind of this little stuff that looks like broccoli florets. Is the choroid plexus streaming out of the interventricular foramen. So it's coming from the lateral ventricle and streaming into the third ventricle. Okay? So what we want to do now is look at this with respect to making the cut. So the first one I want to cut is do the horizontal, because that's the one that we've used the template most of in class, which then defines basal ganglia, the different components of basal ganglia, and the internal capsule. So I'm going to make a cut right through interventricular foramen. I'm going to kind of angle it back here so I get just a little bit of the splenium of the corpus callosum, or the caudalmost part of the corpus callosum.
Similar to cutting like a piece of bread?
Clay, like old Play-Doh clay kind of stuff. Okay, so now put it back together. So what we've cut through is we've cut through the rostrum of the corpus callosum, the fornix, the diencephalon, the splenium of the corpus callosum, and then on back through the occipital lobe. So when we look at this, notice here's that V-shaped structure of the internal capsule coming right to interventricular foramen. And then lateral to that in the V is the lentiform nucleus. And this is a really good image, because what you can see is you can see kind of the difference in colors of the putamen and then the globus pallidus. You can't really see the two divisions of globus pallidus, but you can see the two different parts of putamen and GP. Now, the other thing that you notice on this is that this internal capsule this anterior limb divides off the caudate nucleus, which is in the wall of the lateral ventricle. So here's the lateral ventricle. And the posterior limb divides the lentiform nucleus away from the thalamus. And remember, I told you, you're not going to be able to see specific nuclei in the thalamus. All you really see is just kind of this homogeneous gray area. Okay? If you follow the internal capsule posteriorly, you'll see this large white matter bundle sweeping around like this. These are the visual radiations or geniculocalcarein fibers, or however you want to know them, but coming from the lateral genicular nucleus, which is down under here, and sending those fibers back to primary visual cortex. Okay? If we look at it from the lateral side, here's our lateral sulcus, and that cut went right through the lateral sulcus, and you see in the lateral sulcus, you see this gyrus running back this direction. That's the transverse temporal gyrus of Heschel, or primary auditory cortex. And when we open that up and look inside the lateral sulcus, we see that cortex is folded behind that, and that's the insula. Okay? Insula. Okay?
Where would Wernicke's area be?
On this,
So it's further up, it's not being cut in.
so what you do is you look at this and you say, okay, I'm going to follow the lateral sulcus back, and it's going to kind of end. I'm going to follow superior temporal sulcus back, and it's going to end. Well, all the superior temporal gyrus is area 22, and it's just this caudalmost part of area 22 that's referred to as Wernicke's area. And sometimes it, like I said, it will sweep up into here. This area up here is area 39. Okay? Now, so what I want you to think about is, when we look at this, let's look at it from this side. Look at it from this side, is that the internal capsule is that capsule of, it's just fibers going up and down. There's no cell bodies in there or anything. It's just fibers going up and down. And if we continue and follow it down, some of the fibers that are going down through the internal capsule or out of the cortex into the brainstem are a combination of corticospinal, corticobulbar, and corticopontine fibers. So remember, we have corticospinal fibers going down that will ultimately cross and give us fractionated movement. We have corticobulbar fibers that are distributed bilaterally to cranial neuromotor nuclei. And then we have the corticopontine fibers that are going down to end in pontine nuclei. Okay? So we're going to make another cut a bit inferior to that. And now we're down at the point where we've cut through the rostralmost part of the temporal lobe. We've cut through, let's put it back together. I'm going to lose my brainstem there. Let's put it back together. So we've cut through the caudalmost part of the midbrain. We didn't get the cerebellum. If we pull that away and we look at that orientation, now we see the midbrain, and in the midbrain you can see cerebral peduncles, and then you can see this black or darkened line right here. That's substantia nigra pars compacta. So you can actually see the melanin pigment that shows up in pars compacta for dopamine synthesis. You can actually see that in the brainstem. Okay? Now, I'm going to get rid of him for a minute. When we talked about the limbic system, we talked about input coming into cingulate gyrus, and that the cingulate gyrus continued around the splenium of the corpus callosum to become the perihippocampal gyrus. And then the rostralmost part of the perihippocampal gyrus, this area right here, is named the uncus. I don't know what uncus means, but it's uncus. Now, the uncus marks the position of the amygdala, or the rostralmost part of limbic system structures. And if we look at this, here's the interventricular foramen. The uncus lies just inferior to it in the rostral medialmost part of temporal lobe. Okay? Now, so limbic system information comes flowing in here, and as it flows in here, it sweeps around and comes to perihippocampal gyrus, and then the structure that lies just inside the perihippocampal gyrus. So here's the end of perihippocampal gyrus going into the uncus. If we go just inside that, there's this area right in here, which is the hippocampal formation. And then just at the uncus right here, here's the amygdala. And in a horizontal section, what you notice is that the amygdala lies anterior to the tip of the lateral ventricle, and the hippocampus lies on the medial wall. Okay? So when you look at this ventricular model, what we've done with this is we've made a horizontal cut right through here, so we see the continuity of the lateral ventricle like this. So we just cut through the bottom part of that curvature, okay, with the hippocampus and the amygdala, which are those two limbic system structures. And we'll call them subcortical, even though they're not completely that way, are the subcortical parts of the limbic system that receive all the sensory information that's necessary for consolidation of movements. Remember, or memories. Remember that the hippocampus is for that explicit memory, or the memory of facts, and the amygdala is more the memory of emotions, and primarily more fearful type emotions as opposed to joyful type emotions. Okay, so it's really a protective. It's that part that generates sort of protective responses. All right.
Can you distinguish the cerebral peduncles?
So let's do it this way first. And let me put this up here. Okay? So the primary component of the cerebral peduncles comes out of the posterior limb of the internal capsule and less from the anterior limb. When you think about the internal capsule as fibers that are coming up into the cortex, but there are also fibers that are coming down, and as they continue to go down, they start to collect, kind of like the bell of a funnel coming down to the point of a funnel. And at the point that they travel through this part of the brain, they get to now all be collected as a cerebral peduncle. So the internal capsule, as it continues down, becomes the internal, or the cerebral peduncles. Okay? Now, let's look at the cerebellum a minute. So the cerebellum, here's our cerebellum, and remember we divided it into 3 functional divisions. And we can look at that from the point of view of, here's the superior surface going around to an inferior surface, and we cut right down through the middle, This middle is called the vermis. When you look at the vermis, get away from it. You look at the vermis and then just take the area laterally adjacent to it all the way around, called the perivermal zone. That strip of cerebellum on both sides is referred to as the spinal cerebellum. So that's the part of the cerebellum that has to do with axial musculature, posture, that kind of thing. And it's getting mossy fibers, it's getting climbing fibers, but the primary source of all of that has to do with sort of midline musculature and posture and position. Okay? If you look at the inferior side of this piece, and you look and follow it around, you see kind of this dip right here, and then you see how a little bit of cerebellum's bunched up here. These are called the tonsils, and they are just a landmark